The session, as the name suggests, is about post-winning E. coli, and we have seen over the last, I would say, couple decades, a change on the regulatory usage of antimicrobial, and that, of course, would impact the different types of bacterial pathogens, and E. coli would be one of them, and we have to think more and more about how to use antimicrobial strategically, and it becomes even more important for us to characterize that and use the most advanced diagnostic tools that we have, and combine that with the field experiences with the practitioners that have been dealing with that in the field over the years, and how they can get the most out of the most advanced diagnostic technology. So we would get going with our first speaker, Dr. John Fairbrother. Dr. Fairbrother graduated from the as a Bachelor of Veterinary Science in 1972 from the Faculty of Veterinary Science at the University of Sydney, Australia. And he worked in large animal practice for several years, completed a doctorate in veterinary microbiology at the College of Vet Med at Cornell University in New York in 1981. Then joined the Vet Med school in University of Montreal in Canada, building up a research program on identification, pathogenic mechanisms, and control of and diagnosis of E. coli causing disease in animals. He is going to speak about the whole genome sequencing and how that can help us understanding the change and the phases of pathogenic E. coli in pigs. Round of applause for Dr. Fairbrother. Thank you.
Thank you.
Good. The bottom one is the pointer, and move forward.
Thanks. Thanks very much, Fabio, for the introduction, and thank you to the Leman conference for inviting me to come here to this beautiful city of St. Paul. Fabio was talking about the new technologies, and what's been happening over the last several years is that the whole genome sequencing has become one of the biggest changes in technology in recent years after the PCR. And one of the problems and it can give us a lot of information, but it's still relatively expensive, and what we're trying to do is to show you, the practitioners, just how important it is and how you can use it. So that's a little bit of my take-home message for today. And the other thing is to look at what's been happening over the last 30 years as far as E. coli causing problems in pigs. So the first thing I'm going to do is just very, very quickly, I won't go into details, but I just want to get you aware of the fact that with E. coli, there are four different pathotypes, main pathotypes, which are involved in causing disease in nearly all of the production species, and a whole lot of different diseases are caused by this. I won't go into all of them, but just to say that the ETEC, there's EPEC, XPEC, STEC, and ETEC. And ETEC is probably the most important as far as we're concerned today. Now, the other thing I want to say is that the identification of these different pathotypes and the diagnosis of them is based on the presence of virulence factors, for which there are virulence genes. And so, and I've noted some of the genes here, but I'm not going to go into the details. You can look at the slides for that. But the point here is that the diagnosis of these diseases, of the identification of these E. coli, is based on the presence of these virulence genes. And so there are virulence genes which define each of the four pathotypes. What I'm showing you here is that, for the diagnosis of these cases, what we do is, once again, as I said, is based on a PCR, but we go in and we do an initial culture, broth culture of the sample, and we do an initial PCR before we even look at colonies. And if it's negative, then we stop. If it's positive, then we say, okay, we're going to look at the colonies, and we look at the colonies for these defining E. coli, these defining virulence genes. And this gives, when we find these, in the colonies with a certain pattern profiles, and we say, okay, this is a virulent E. coli. The other thing that you can do, and I don't know if you, hopefully you can see this on the slide, down the bottom, is that once it's identified by PCR, some people will do a MALDI-TOF to be able to say, yeah, this really is an E. coli. And most practitioners, most diagnostic labs will also do a antibiotic sensitivity testing. And the last thing that some labs will do, but mostly not, is looking at phylogenetic analysis for clonal, seeing if you have clones or clonal lineages there. And most of these tests are fairly complicated, more complicated and more expensive to do. So we'll talk about those as we go along. I'm just showing you here, and once again, it's not to necessarily go into the details, but if you look over to the left, you have the 4 different pathotypes. If you look at the next column, it's what we call the pathovirotypes. And here, what this means is that instead of just saying ETEC, we'll say ETEC F4 or ETEC F18, and this is the information, and I'll go back to that, but in the next column, it's the different virotypes that we find. And then you'll see there are a whole lot of different virotypes. And probably at the practitioner level, it's not that important to know the difference between them. But what is important to know is, for example, that the strain has F4 or F18, so you know if you're going on a vaccine strategy. So usually when we read this out, and this goes to the practitioner on the report, the most important thing will be that it's an ETEC F4 or an ETEC F18. Over to the right, I'm showing the ST, or the sequence type, and the reason for this is data coming from all of which is published worldwide. And what I'm showing is that some labs do show they'll do the sequence typing, and this is a test which it's a bit more expensive to do, so it's mostly in reference labs, but there is some information already on that. Okay, so we'll get on to our results of what we've seen over the last, in this case, we do surveillance. We've been doing surveillance since 2008. And when we do our annual report, it's for the last 5 years. So what I'm showing here is the report between up to 2020. We have the results for the rest, but it hasn't been validated yet by the Minister of Agriculture. So what I'm showing here at first is the trends we see in the pathotypes. And the most important here to note for you is if you look up the top of the line, the red line is the ETECs, and the ETEC F4, I'm sorry, the pathovirotypes. And in our case, this is the most important ones. Now, if you go elsewhere in the world, if you go here in the States, it may be slightly different. If you go into Europe, it's going to be more the ETEC F18. Now you see in our case, we do see them, and there's an increase. This is the orange line down the bottom, and we're seeing an increase with time, but it's not as much as the ETEC F4. So now if you look down into these ETEC F4 and find out what the virotypes are, what you'll see is that you see a blue line up the top towards the left, and there's an orange line which as you go towards the right, start to drop. So there's been a decrease in prevalence over the last 5 years. These are the LT STA STB F4 and the LT STB F4. At the same time, you see a red line going, which started low at the beginning of the time period and has been increasing with time. These are the STA STB F4, and so this is the one which is predominant now. Okay. I'll come back to this. Keep this in mind, because once we do the whole genome sequencing, this will become much, much more interesting. The other thing I want to show with the antimicrobial resistance results is that if you look up the top, the two graphs at the top represent the strains which you saw on the lines which are going down with time, and these ones are. What we're looking at here is enrofloxacin resistance, and yellow is intermediate resistance as well. So you'll see that the majority of these are the STA, STB, LT are all resistant or intermediate. The ones on the right up the top are the LT, STB, and half of those are resistant. Now, if you look down the bottom, the new one which is coming in is not, there's no resistance to enrofloxacin. If you look now at the multi-drug resistance, the same up the top there, to the left is the LT-STA, STB, F4, and what I'll point out, well, first of all, you'll notice that the green down the bottom is those which are not multi-resistant. That is, they'd be sensitive to all except for 2 or less classes of antibiotics. But what you see up the top, the red one is where they're resistant to 7 or 8 of the 8 classes which are tested. So that's quite a panicking sort of thing to see. In other words, there's not much you can use to treat these guys. These ones which are enrofloxacin resistant are also resistant to, sometimes they're extensively resistant to a whole lot of antibiotics. Now, if you look down the bottom, the new one that's coming on is, once again, there's not much green there either, so there's not many that are not multi-drug resistant. And the interesting to note is with the red ones, we didn't see any in the beginning, but now we're starting to see some of those which are resistant to, they're red, so they're resistant to 7 or 8 of the antimicrobials. The other interesting thing is that the two at the top, which are in the enrofloxacin resistant group, are, if you look at the last 2 years, 2019, 2020, there's no red there. So maybe this is showing this after 2018 when we start to have a change in the antimicrobials which are being used. You're starting to see less resistance, less resistance, maybe. So what we did, we looked at all these strains by pulse field gel electrophoresis, which is a phylogenetic testing for looking for the clonal types. And down the bottom you see the circle there shows that there is a clonal cluster there, but it's not discriminate enough to let us say any more than that. So that's where we stop with the tests, the traditional tests. And I just want to point out, we talked about clones. I just want to make the point that in the intestine, the E. coli in the intestine of the pig, there are many, many clones. And I put them in colors which represent the one up, the pathogenic ones. So the minority, well, go down the bottom, the commensals, that's the majority of what you see, but they're made up of different clones. And you'll see the pathogenic ones which are put in green up the top. So they're a minority one, and this is the one we're going to be talking about more. And those which are potentially pathogenic are those which are, have virulence genes, but they're not enough to, we don't know what the virulence genes do. And I've just made the point over to the left that many of these, or most of these, will have plasmids, which are able to be transmitted between the clones. Now, what I'm going to introduce a little bit now, and this is going to help us as we move along into the whole genome sequencing, is that over to the left you'll see that we're talking now about persistent pathogenic clones. Persistent because they stay around for a long time, and this is becoming part of the problem. If you look over to the left, you'll see that I've listed certain of the antibiotics, and to show that in these clones you'll have resistance genes, but also mutations for resistance. And so mutations for resistance in the chromosome, examples of that are colistin and the enrofloxacin. The enrofloxacin we've just been talking about. And if you look down the bottom at the left, you see that on the plasmid you have other resistance genes which are present on the plasmids, such as for ceftiofur, tetracycline, etc. So that's one type of gene which is present on the plasmid. If you look over to the right, you have the virulence genes I've mentioned, and these are also present on these same plasmids, and also fitness genes. Now, these fitness genes are genes which allow the bacteria to persist in the environment. And these we're getting to know more about, and they're very important in not the pathogenicity, but the ability of these bacteria to hang around for a long time. So keep this in mind. We're going to come back to this a little bit later in the talk. So the question now is, how can we go further using whole genome sequencing as this new technology? And I guess you probably don't see a lot of that, but and unfortunately I don't either. But what I'm trying to say here is that whole genome sequencing, it's a very large thing. So there are a lot of things you can do with it, especially with bacteria. And up the top, what I'm showing is that first thing is you can look simply for the presence of genes, virulence genes, example, antimicrobial resistance genes, genes which tell us the serotype, genes which tell us there's an E. coli, genes which are present on plasmids, etc. So you can just simply use it for looking at this. Second thing you can do is to look at clonal relationship, look at whether the strains are related, and here there are different analysis such as using the SNPs or using the MLST. Now you can use the traditional MLST with using the same genes that we use there, but also you can use the core genome or the whole genome MLST, which involves now more than 1,000 genes, so it's very, very, very discriminate. Well, finally, down the bottom, what I'm saying is that you can follow outbreaks, and this you can do using phylogenetic trees, such as SNP trees, which now brings us to, I hope, no. So I'll just go through very quickly to show you. In our case, we use the Illumina platform. There are several others. So in our case, it's the Illumina one, and we use you can use MySeq or ISeq, and we use both. But we use mostly the ISeq now. ISeq, we can do 10 isolates at a time, which means that you don't have to. With MySeq, it's 30, so you have to wait till you have 30 isolates. So in this case, we can run this. We get a strain come in, an isolate come in. We can do the test within the next couple of days, and they can have the result coming out. So I won't go into details of the technical details. It's just showing that we use this to you have your reads, you reassemble, and then use either databases which are publicly accessible on the internet, or software that is now more and more available. So this is just showing the pipeline that we use, and up the top I'm showing you the whole lot of different, there's between 8 and 10 different databases that we go in to get these results. And then down the bottom I'm showing that we're making the phylogenetic trees. So obviously you're not going to, what I'm just showing you here, in case you don't see it, the details, I won't go into all the details, but just showing you this is the report that we send out when we do a whole genome sequencing, and basically showing you the information they get, and down the bottom we're looking at something like 700 virulence genes now. So we put these into about 10 different classes of functional classes of virulence. We do a tree, and in this case, phylogenetic tree. In this case, with the strain that we're looking at, we take about between 10 and 20 isolates, strains coming from representative of in this case in pigs in our geographical area, and we compare this coming from different pathogenicities, and in this case we're looking at we compare this with the strain that we're testing. We also look at antimicrobial resistance by class, and I just want to say we can predict you can predict the resistance using the class, and in most cases it is fairly correlates very well with the phenotypic. In some it doesn't, but it's getting better as we go along as we have more databases available to us. Okay, so now I'm getting on to the results and of what we've seen over the last 30 years. And I'm not going into detail. All I want you to see on this one is this is a circular SNP tree. And this is the SNPs are the ones that people use most to be able to distinguish the strains. There's about 150 strains there. The only thing I want to show you there, this is 30 years of strains. And what I want to show you is that over the 30 years, there are just a few clones which are predominant over this time. So it's not a matter of having a whole lot of heterogeneous E. coli there, it's a matter of having clones which have been present over the years. And I'm going to show you more detail of that in a second. Just to zoom in a little bit, you'll see that this is one of the clones, the A1, and there are 2 points I want to make there. If you look at the outside of the circle, you'll see that there are little triangles, and what these triangles represent are different mutations which result in the resistance to fluoroquinolones. And the more of these mutations you have, the more likely they're going to be resistant. And in this case, they have mostly 3, 4, or 5 mutations, so this correlates with what we've seen, that they are quite resistant. The other thing which you probably won't see from where you are, but in the first circle of color coming underneath the triangles, you'll see that we're looking at the different virotypes, and what's happening is that in this one clone, you have both STA, STB-LT, and LT-STB. So you have 2 virotypes within the same clone. And this is what we saw in the initial graph. Now, I've summarized what we've seen over the last 30 years, and this is really, really, really interesting. I'll take you down. If you go up to the top, you'll see that in the beginning, and this was in 1990, you saw that there was an initial clonal lineage, or it's based on the ST, which was present, as ST90, and it was very heterogeneous as far as the serotypes which were present. Not so much the clones. But then what happened in 1995 or about, you had a totally new clonal lineage which appeared and has been present all along up till now. And at the same time, all of the isolates in this clonal lineage have 5 more virulence or fitness genes than what was present before. And so this is the whole genome sequencing which has showed us this. And there's a higher risk of mortality in these. And those who were around practicing in 1995, any in the room will remember that at that time, that was when we started to see all through North America a form of E. coli of post-weaning diarrhea, which was much more severe. And you had this even sudden deaths going right out into the growing period. And in the beginning, people misdiagnosed this as strep suis or other, and finally by accident we discovered this was E. coli. So, and this was relatively panicking for the practitioners, except that at the time there was still enough antibiotics that could be used so that they started to vaccinate, but also there were still a lot of use of antibiotics because there was still a lot of sensitivity. Now, it's clonal, so in the beginning you had 2 clones which are called A2 and 3, and then when we get to fast forwarding here to 2013, 2014, you get all of a sudden you get this A1 which appeared and has we see up till now. It's still present. Now you look down the bottom, and what's happening is that there is a new clonal lineage, this ST772, which is an O23, different clone, and it's one clone for the moment, and it has one virulence gene, not the same virulence genes as was present in the previous one, but one virulence gene more than what we saw, say, 20 years ago. So now if you go back to the data that we had by PCR, you'll see that, in fact, this new A1 is what is represented by what we saw before, these 2 lines, which were the isolates which were STA, STB-LT, or STB-LT, F4, and what we're seeing is that there's a decrease in prevalence now. Whereas the new one is this STA, STB, and this is the ST772. So that's kind of neat. Now, to go even further, what happened was that one of the beauties of using whole genome sequencing is that you can look in databases and look at strains, sequences which are already published. So what we did was we looked at sequences of which had been published from a study here in the States, and lo and behold, the same clone, which is present in Quebec, is also present and was predominant in the collection of isolates which were in the publication, in most of the pig-producing states of here in the United States. So, in other words, it was present everywhere in the United States, as well as at least in Canada and probably in the other provinces as well. It's just we don't follow in the other provinces. So it's pretty widespread. And if you look now, you'll see that we looked over in 2013. This is where we first started to see some cases. In Quebec, this is the pig-producing map of the pig-producing area of Quebec. 2014, you start to see more cases. 2015, it's emerged. So we're seeing it pretty much in more areas. And then in 2016, it's everywhere in all pig-producing areas of Quebec. And so if you look at, this is simply, we did a study where we put GPSs on veterinarians and on transport trucks for the pigs, which go between 10 different piggeries in a network, and an abattoir which is associated, and you can just see how much movement there is between the farms and the abattoir. So this can help you to imagine just how these E. coli are able to get around now in between, in knowing how much movement there is of pigs between the different provinces of Canada, Canada, and the different states of the United States, just how much movement there is and how this has got around. And the other thing is we've shown that it persists for at least 6 months, and probably much more than that, but this is as far as we've been able to evaluate it on the same farm. So it persists, it hangs around. So this brings us back to the same diagram I showed you a bit earlier, the bacteria, the clone, and now you see that I've changed the name. It's called a high-risk clone. And see, this is the new definition of, and this is seen as much in other species. In humans, it's there's one which is associated with uropathogenic disease, which is worldwide. So so this is basically what we're seeing here. These are isolates which clones which are able to persist. They're epidemic. They're easily transmitted. They're more pathogenic. And so, how do they get that way? One of the main reasons is that over the left you have the different antimicrobials which have been used in different ways, and which drive the presence of the virulence genes, of the mutations. And at the same time, you have these plasmids which are being pushed and driven, and at the same time they have virulence genes, and they have fitness genes on them as well. So that these things become more pathogenic and they become more persistent in the environment. So this is where we are at the moment. So, in answer to what the question I asked at the beginning, genomic characterization has demonstrated the emergence of a high-risk clone of pathogenic E. coli associated with more severe post-weaning diarrhea in pigs throughout North America, and will permit now in the future a more rapid and accurate monitoring for the potential emergence of new high-risk clones. And the point I can make there is that with the new ST772, so far we have one clone. So far well, I can't say whether it's easily transmissible because I don't have too much data for what's happening in the United States. But at least in our case, it is the predominant one at the moment. So I'm going to finished there. I'll just go back. What I want to say at the end is that I've, in thinking about reflecting on all of this, just how we can show what is the added value for a practitioner to be able to use the whole genome sequencing, and I've divided up into indirect and direct usefulness for the veterinarian. Indirect just means that what's happening is, as in the case of what I've just shown you, this is data which is coming from research and from our monitoring surveillance, and it is indirect because it also helps you, the practitioners, to know what's going on. But direct is that what I'm trying to convince you of here is that there are direct ways of you using, once you see that these are important, is to make more use of this, of the whole genome sequencing as a diagnostic test. And I'm just going to finish up by saying that I would advocate that this is an integrated thing, that what we should be doing is we keep going with our surveillance of, as we do with the PCR and antimicrobial resistance, so we see what the trends are, and when we see a change, then we can go zero in and test certain isolates by whole genome sequencing to be able to say either to confirm or not whether we have new high-risk clones coming in. And then when we have that, we can you can go in and you can look at which preventive or biosecurity or whatever the approach that you can use based on the whole genome sequencing results. So I'll finish there, and thanks very much.
So we may have time for one question for Dr. Fairbrother. Okay, question over there. You have a mic for? Oh, mic is coming on there. Yeah, there you go.
Thank you, John. I'm just wondering if you also tried to detect the toxins produced by these new clones, aside from just the O and H?
Yeah, that's amongst the, you probably didn't see it, but it's amongst all the virulence genes we're doing. Yeah, always. Okay. Always.
Yeah.
So the known toxins, simply by the presence of the genes. We don't go testing phenotypically, but we're always STA, STB, LT, and STX1 and 2. Oh, and with the whole genome, you also got all the other toxins that could be present as well. Yes.
Okay. Okay. So thank you again, Dr. Fairbrother, and we will move to the next speaker. Thank you.